Hello and welcome to another video in our course on cell biology and genetics. In this video, we're going to talk about translation. So what are you going to learn in this video? First, I'm going to explain what translation is and how it works. I'm going to explain what codons are and why ribosomes are so important for translation. We are also going to talk about tRNA and post-translational modifications. After watching this video, you will also understand the terms initiator tRNA, rRNA, A, P, and E site on the ribosome, anticodon, amino-acyl-tRNA-synthetase, initiation complex, start codon, and stop codon. By the way, you can find written notes on what we're going to discuss today at medicinecanbeeasy.com. The link is also in the description. Okay, so now let's explain what translation is. Translation is the second step of gene expression, the process in which the information in the gene is used to synthesize a product, usually a protein. The first step was transcription, the synthesis of RNA from DNA. Translation is the process of synthesizing a protein from the transcribed RNA, mRNA. Before we look at the process in detail, let's talk about all the necessary translation components and terminology to understand the process. We'll start with codons. A codon is the sequence of 3 nucleotides in the mRNA molecule. One codon specifies one amino acid. As you can see here, uracil cytosine guanine specifies serine, adenine cytosine asparagine, uracil guanine cytosine cysteine, and guanine adenine guanine glutamic acid. Next, we have the ribosome. A ribosome is an organelle made of small ribosomal proteins and RNA molecules called ribosomal RNAs, rRNAs. It is composed of 2 subunits, a large subunit and a small subunit that fit together. A ribosome contains 3 sites: amino-acyl site, A site, peptidyl site, B site, and exit site, E site. We are going to talk about them later in this video. Finally, the tRNA. tRNAs, transfer RNAs, are small RNA molecules that transfer amino acids to the ribosome. They have a very specific structure because they contain 4 short segments that are double helical and make the molecule look like a clover leaf. 2 of these regions are especially important. The first one is the anticodon, 3 nucleotides that bind through base pairing to a complementary codon in the mRNA molecule. For example, a guanine cytosine codon in the mRNA specifying alanine requires a complementary cytosine guanine anticodon. The second one is the amino-acid-accepting arm, where the amino acid gets attached to the tRNA. The attachment of the amino acid to the tRNA depends on enzymes called amino-acyl-tRNA-synthetases. There is a different synthetase enzyme for each amino acid. Each synthetase recognizes a particular amino acid, as well as all the tRNAs that accept that amino acid. Now that we understand all that, we are ready to take a look at the major steps of translation. After the mRNA molecule is synthesized during transcription, it travels to the cytoplasm. Here, several components bind to the mRNA: the small ribosomal subunit, initiation factors, and the so-called initiator tRNA. Initiator tRNA is a special tRNA that always carries the amino acid methionine and is required to initiate translation. These form the initiation complex that binds to the gap at the 5' end of the mRNA. This initiation complex then moves along the mRNA to find the adenine-uracil-guanine codon, specifying methionine, so that the initiator tRNA can bind to it. For that reason, the adenine-uracil-guanine codon is called the start codon. Once found, tRNA pairs with the codon, the initiation factors are released, and the large ribosomal subunit joins the small one. With methionine as the first amino acid in the protein chain, although it can be removed later, the mRNA is pulled through the ribosome in the 5' to 3' direction, and its nucleotide sequence is read in groups of 3 in codons, and amino acids are gradually added to the polypeptide chain. To understand the process in more detail, we need to look at the ribosome and its 3 special sites that we mentioned earlier: aminoacyl-A site, peptidyl-P site, and exit-E site. First, tRNA with the amino acid enters the A site of the ribosome, where it pairs with the complementary codon. The amino acid bound to the tRNA is then linked to the peptide chain in the P site. After that, the large ribosomal subunit moves forward, and the tRNA that was in the P site shifts to the E site, where it is ejected. In this way, a protein chain is gradually synthesized. Translation ends when the ribosome reaches the so-called stop codon. Stop codons, uracil-adenine-adenine, uracil-adenine-guanine, or uracil-guanine-adenine, do not specify an amino acid and only give a signal to the ribosome to stop translation. Proteins, known as release factors, then bind to the stop codon, and the completed protein chain is released, together with mRNA, and the 2 ribosomal subunits dissociate. Once the synthesized protein leaves the ribosome, it can be modified in different ways. These modifications are called post-translational modifications, and they can include processes like methylation, adding a methyl group, glycosylation, phosphorylation, etc. These modifications are often needed for the protein to become fully functional. Okay, so now let's summarize what we've learned today. First, we said that translation is a process of synthesizing a protein from the transcribed RNA. We also talked about codons, sequences of 3 nucleotides that specify amino acids, and ribosomes that have 2 subunits, a large and a small subunit, and 3 functional sites, A, P, and E site. Then we mentioned tRNAs, which transfer amino acids to the ribosome, and we talked about their specific structure with the anticodon, which binds to a complementary mRNA codon, and the amino-acid-accepting arm, which attaches the amino acid. We said the enzymes responsible for attaching the amino acid to the tRNA are the aminoacyl-tRNA-synthetase. Then we said that in order to initiate translation, the small ribosomal subunit, together with the initiation factors and the initiator tRNA, forms the initiation complex, which binds to the 5' end of the mRNA. Once it finds the adenine-uracil-guanine codon specifying methionine, tRNA pairs with it, initiation factors are released, and the large ribosomal subunit joins the small one. A protein chain is then synthesized, and translation stops when the ribosome reaches a stop codon, which gives a signal to the ribosome to stop translation. After the protein is released, post-translational modifications often take place for the protein to become fully functional. Okay? Great. So this is it for translation. Thank you so much for watching. If you found this video helpful and you'd like to watch more videos on cell biology and genetics or medicine in general, please subscribe. Thank you again and see you soon.